So this is a chapter on gene therapy, and just quickly, since most of you are here, today is 7th April, and so your midterm, as you know, is scheduled for 14th April, Thursday, and the syllabi is cumulative, and it will be just like on the pattern of quiz, and there won't be any class, but I will be here, in my office hours. So multiple choice questions, just like quiz 1 and quiz 2, and the course is cumulative. Any questions about the midterm? Okay. So if there are no questions, okay, then we are. Gene therapy, the word therapy means treatment, and the gene therapy means treatment by gene, treatment by DNA. So DNA is the pharmaceutical mode to cure sickness. So why do we need to do this? Because gene therapy is needed in the situations where a particular gene has gone alteration in its nucleotide, and the problem is that it's very hard to rectify a gene that has altered nucleotide, because an altered nucleotide will give rise to altered RNA or mRNA and then to altered protein. So I want to teach this class using a model, a disease model of sickle cell anemia. Okay. So this is the slide. So from here, you can study from DNA to protein structure to protein function. So what you see here is a normal gene for hemoglobin, which has, so focus on this triple nucleotide, GAG, CTC. GAG codes for glutamic acid, and if you go to the protein amino acid table, glutamic acid is a negatively charged amino acid. And because of the missense mutation, the A, which is a purine, it gets mutated to T, which is thymine. So from purine to pyrimidine, this is transversion, and then this is missense mutation because it's still coding for an amino acid, and amino acid is valine. So proteins are very sensitive to amino acid, particularly their side chains. Side chains offer them a special chemical property. So this is a negatively charged amino acid, and this becomes hydrophobic amino acid. So with negatively charged amino acid on the surface of hemoglobin has the potential to interact with various solution buffers in the cell, or but when it becomes hydrophobic, this protein can collapse, right? Because if you compare, the glutamic acid has a longer side chain, right? And valine has a very shorter side chain. So there's a shock to the protein. And because of this, the spherical hemoglobin, it becomes crippled, crescent shape, crippled shape, right? So what is the consequence of this? The consequence of this, hemoglobin loses its capacity to bind to oxygen or carbon dioxide. So such patients will need transfusion. And in developed countries like US, patient live, could depending upon other clinical situations and other health conditions. Sickle cell anemia, anemic patient could live for long time, but in developing countries or in poor countries where more than treatment, you need to maintain the clinical necessity of such individuals. There, the fertility rate is high and at much younger rate. So this is, and another thing interesting is that sickle cell anemia, that's one of the hypotheses, is that that sickle cell anemia mainly developed in those areas of the world where malaria was endemic, right? So, so because malarial parasite, it enters into RBC, and that's the house where they live to grow. So so this mutation, from evolutionary point of view or environment point of view, it's possible that human genome or human genetics developed a mechanism to become resistant to malarial infection. So you will see that that sickle cell anemia in early days when it was started. Nowadays people travel, so many of the diseases are in every part of the world. But sickle cell anemia was mainly located at tropical countries where the incidence of malaria was high. So this was a nature mechanism, because malarial parasite can do nothing to a sickle cell anemia, but a normal person, malaria can do many things. So this was another, so this is, I went off the topic. So coming back, what you see that a single nucleotide has altered the shape of the protein in a very dramatic way. So the clinical management of such patient is that you take blood transfusion whenever the blood is not healthy. But that's a very temporary solution on ongoing basis, right? But the permanent solution would be to change, to edit or change thymine back to adenine, such that if normal people have GAG, the sickle cell anemic genotype also has GAG. Now, broadly, there are two strategies to go for gene therapy. Number one is gene insertion, and number two is gene editing, right? In this class, we will mainly talk about gene insertion. Gene editing we'll talk in the next class by learning about CRISPR. So, so now what is gene insertion? So let's say this is a cell, and within the cell, there is a nucleus, right? And within the nucleus, of course, there is DNA, and one of the chromosome is having DNA gene that is mutated. So gene insertion means few things, but before I go into gene insertion, in light of technology, growing technology, there are various ways we can try to attempt to treat this. One is mRNA. If you put mRNA if you inject healthy mRNA, which produces normal hemoglobin, then it's possible that the normal hemoglobin will rescue the disease hemoglobin, right? So, but mRNA has shorter life. So how many times will you inject mRNA? Giving mRNA as a vaccine that we got for COVID-19 is different, because you are developing the immunity, enriching the memory cells. memory B cells to produce, or T cells to produce antibody. That's a different situation because the memory cells are permanent if the vaccine is, if the person is immunocompetent. So mRNA could be a strategy to treat sickle cell anemia patient or an individual. Second would be to insert or to inject or insert normal DNA. Now, how do you inject, right, normal DNA? And what will happen? This normal DNA would go inside the cell and would go inside the nucleus, and this will rescue the diseased the disease gene. So now, when you are inserting something foreign DNA, then you need a vehicle, right? You need a vehicle, and that vehicle here is a vector, right? A vector we have discussed in, I think, second or third chapter, various plasmid, cosmid, and bacteriophages, TI plasmid, many things. So vehicle. So number one is mRNA. The second is to inject normal DNA. Now this vector can be of two types. Number one, it could be retrovirus based, or second, it would be adenovirus based. Now, for both, the strategy will be very different. For retrovirus is an RNA virus. So here you would need the help of the RNA virus that would RNA virus vector, retrovirus vector that would, and retroviruses, they have capacity to insert into the chromosomes. So these will insert into the chromosome. Adenovirus is a DNA virus. Adenovirus is a DNA virus. So again, the genes would be the normal hemoglobin gene will be cloned to retrovirus or lentivirus or adenovirus, and they will be taken into the cells. Now, which cells would you prefer to do this? So the preferable cell here would be bone marrow cells, because bone marrow cells are the cells, multipotent cells that can give rise to a lineage of cells which will develop into RBC, right? So many things to work upon, right? So one is the best vector, then the best cells, and then you need a best lab to do this experiment. And then once you get the best lab, you need the best hand doctor, surgeon, oncologist, or not oncologist in this case, but hematologist, because it's a blood-related disease. So hematologist and a surgeon who can do a bone marrow transplant, right? And the other strategy would be to have stem cells. So get the patient stem cells from bone marrow, get the bone marrow, then purify or extract the stem cells, and then rectify the mutation in the stem cells, and then put it back, right? So as I mentioned to you, I'm teaching you in a in a different way because I'm taking a most translational example, right? Otherwise, I can teach you what is gene therapy, what is insertional gene therapy, what is uh, gene editing, and done. But I'm talking you with the example and presenting to you the clinical picture, biochemical picture, molecular biology picture, so that you develop the ability to think in that way. So when you get into exam mode, I'm not talking about my exam. The exam for this course is pretty straightforward. But if you're taking exams which would require your analytical thinking and connecting many dots, presenting, so you need to think holistically from all the angles, right? So that's what our teacher taught us when I was an undergrad like you. Then that when you look at a question, try to uh, look around, look at the bigger picture of the question, then narrow down to the question. Because in our days, the questions were not multiple choice questions, and we didn't get 2 attempts. We got long questions, and in long questions, you had to you had to write very systematically from A to B, B to C. You cannot. So there are good and bad to both the things, advantages, disadvantages. Uh, but we are from the time when we used to get long answer, short answer question, one line questions. So when you're writing, you're thinking, right? And you're thinking long as compared to when you're taking a multiple choice question, just 1 minute, and then you have to answer. So you, in a fraction of seconds, you have to think about the question, answer it, and the chances of getting right is more, right? You're getting 2 attempts, so you have more chances as compared to us. We have, if the foundation of writing goes wrong, then the whole question will be wrong. And one question would be like 20 points or 10 points. So 10 point went down the waste. But if you in multiple choice questions, you make one mistake, is just 1 point gone. So both disadvantages and advantages. So my professors, my teachers told me that think from all the sides, and as a biologist, because our body is it works on teamwork from various systems. So if one thing goes bad, the other thing will go bad for sure. So anyway, that's the message I wanted to give you using this slide. That bottom line is there are two modes of gene therapy. One is insertional, and one is gene editing. Gene editing, we will take it up on next. Today's Thursday, next Tuesday. And today we will only talk about insertional. So essentially, I taught what I had to teach, but now I will go through the slides. So what is gene therapy? Gene therapy is a mode of therapeutic strategy to treat those diseases which are affected by alteration in the sequence of nucleotide in the DNA. And the function of DNA is determined by the protein it codes for, right? So DNA, so here comes the central dogma of biology, that if the DNA is wrong, then the RNA will be wrong. And if RNA is wrong, then protein will be wrong. So if the protein is wrong, then if one of the protein is very essential biomolecule, if that is wrong, then the outcome of cellular functions will be wrong. So for example, if hemoglobin is mutated, then the efficiency to hold oxygen and exchange carbon dioxide is compromised. So a patient will develop a lot of breathlessness and have to go to dialysis or blood transfusion. And so a good care, a good therapy will only increase the quality of life, right? And historically, the first gene that was targeted for therapy was adenosine diaminase. This is an enzyme that is involved in the biosynthesis of a nucleotide. So if this enzyme is mutated, then the nucleotide synthesis is compromised, and then these people will exhibit a very severe kind of immunodeficiency that is called as severe combined um, immunodeficiency, severe combined. So when adenosine deaminase is used as a gene therapy modality. So, but this was the first gene therapy um, institution. And I already sort of indicated to you that one way is to collect the bone marrow cells. From bone marrow cells, purify the stem cells. How will you purify the stem cells? Stem cells will have a marker, a CD marker, maybe CD34. And nowadays, because of new technology, there will be more markers. So using CD34, you extract the hematopoietic lineage cells or stem cells, and then you grow this in a Petri plate and add, not edit, but add lentiviral vector containing normal hemoglobin or adenoviral vector, and then stabilize these cells, make this transformation, this new cells, genetically modified cells, to be growing and healthy, and then put back into the patient's body. It's doable, but the challenge is the contamination. There are so many deaths in emergency room because of infection. It's very high. Why? Because they have very slow growing bacteria that is multi-antibiotic resistance. And these slow growing antibacteria may not show its effect in the early stage, but it makes patients very sick after a few weeks. So if these cells, if they get infected by slowly dividing bacteria, then these bacteria come into full action inside patient body, and then the experiment fails. So that's, but as the time is progressing, there are very sophisticated equipment where less in a more sterile condition, where the chances of these experiments to work is more, right? So this is using retrovirus vector or adenovirus vector. These cells are genetically transformed because they are now having the gene that will express normal hemoglobin. So, what are the broad types of gene therapy? One is autologous, and that is allogenic. In autologous, the cells from the patient is taken out and treated in the lab. Treated means genetically manipulated, and this cell is again back. So the patient's cells are being used in autologous. In allogenic, cord blood cells are taken from the placenta or from the cord, and then these cells have the potency of a stem cell, and these cells are grown, and then they are naive stem cells, and they are genetically manipulated, and then it is injected into the patient's body or bone marrow. So two types. Autologous means patient and patient cells are genetically modified. Here, somebody else's, like cord blood cells. It may not be from the same patient. So two types, autologous and allogenic. Then the approach could be two types: ex vivo gene therapy and in vivo gene therapy. In ex vivo, the cells are taken out from the body, treated in the lab, by retrovirus or adenovirus. The genes are fixed, and then it is injected into the patient body. So this is ex vivo, is when you take the cells out into the lab. In vivo therapy means when it is directly given into the body. The genes are directly injected. So it could be injected into spinal cord, into bone marrow. So in vivo versus ex vivo is the approach. And then that could be two overall type of problems. There could be problems with, there could be problem in somatic cells, right? Let's say in cancer, some genes are mutated, then those cells, so let's say if somebody has stomach cancer, then those cells will be taken out, chemotherapeutically treated, and if there are mutation, it will be fixed and then put back into the system. Patient's body, I meant. If there is a genetic diseases, for example, in sex-linked diseases like color blindness or night blindness, if the genes have to be rectified, then then the the experiments have to be done. The genetically modification or manipulation has to be done either in sperm or in egg, depends where the gene, the disease gene is coming from. So one is germline gene therapy, which is done in patients' gametes, and other is somatic therapy, where it is done in let's say skin cells if the patient is having melanoma, or mm, in stomach cancer or liver cancer, depends. Now, a method of gene therapy is, it could be uh, injected by needle or micro injections. Electroporation is through using electricity. The DNA is pushed into the cells on the basis of electrical force, or a gene gun technology. So these are physical ways to, to basically pierce the cell membrane and then inject the DNA. It could be retroviral, it could be adenoviral. The second is chemical method. Calcium phosphate, DAE dextran liposomes, they can enter the cells. They have the property to make the cells more porous. If you have done molecular biology experiment, then if you have done transformation, then the plasmid is inserted into the bacteria. Plasmid is inserted into the bacteria through calcium chloride treatment. So here, calcium phosphate can be used to make the cells more permeable, and then let the this DNA go. Second is liposomes. So what are liposomes? Liposomes are small sac-like structure made up of bilipid layer. So liposomes have advantage. So let's say if we want to deliver the gene into a kidney cells, or we want to deliver the gene into the skin cells. Now, the membrane composition of a fibroblast, the membrane composition of an epithelial cells, whether they are in kidney or skin, will be very different. So we can design specific liposomes depending upon what cells to target. So liposomes are lipid bags, like bilayer, and they have a sphere, and this DNA, these nucleotides or these DNA will be encapsulated in this lipid bag, small bag, and they can be through chemical or through independently, they can go into go and merge with the cell, and then the DNA could be delivered. So as I mentioned to you, the DNA could be that the DNA has to be targeted into the right cells, so it could be done through viruses. So why viruses? Because viruses have the unique ability to enter into human cells using receptor. So retrovirus, adenovirus, they are the most preferred vectors that can be used to deliver the target gene. And the most common, the most popular is retrovirus vectors for gene delivery. Why? Because they can integrate into the human chromosomes. So they can generate permanent change into the cells, and they can keep expressing the protein as compared to adenovirus, which don't um, integrate with the chromosome. Okay, so two ways to deliver gene, and of course liposomes and plasmid DNA also. So plasmid DNA is less stable as compared to retrovirus, adenovirus. Now, when there is positive side, there are also negative sides. It's very hard to control these viruses when inside the cell. They can integrate randomly to many places and they can give rise to secondary mutations. So it is very essential to characterize those cells, right? When you're doing the experiment inside with the patient's cells, and when you're selecting, so selecting the right cells is very important, right? So let me show you, there is a company, um, there's a company that is, there is a company, let me bring it to you. Yeah, there is a company, biotech company. Yeah, this is called as BioNano, and they have a workflow. I'm sorry, and where they can take blood cells or cells that has been treated in the lab, and they would look at the complete sequence and do all the analysis before putting into the patient's body to make sure there are no secondary mutations. Right? There are no secondary mutations. Also, you know, um, there are some companies which are developing. They have various. In the pipeline, they have X-linked retinitis pigmentosa, chromatopsia, optogenetics, age-related macular degeneration, non-syndromic hearing loss and deafness. So this is a company which has many gene therapy in pipeline, many gene therapy target in the pipeline. So what is the general strategy? The general strategy is, I can briefly mention to you that viruses, they have good genes, bad genes. Bad genes are those genes which can cause disease, which can kill human cells, and help virus to become pathogenic. So for example, in retrovirus, so what is the example of a retrovirus is HIV. So this HIV, the most damaging genes are GAG genes, pol genes, and envelope genes. These three genes, they help the virus to enter into human body and then replicate. But these three genes, which are not, which are injurious to human health or human physiology, these genes are removed, and instead a new gene is cloned, which could be for hemoglobin, and then retrovirus can be generated, and this can be put back. So here, once this molecular biology experiment for cloning the gene is very standard. The the problem is, the challenge is rather to select those healthy cells which are not having any other mutations, but they are expressing normal human hemoglobin. So you take the patient cells, transduce the cells with viruses, retroviruses with healthy genes, select for those cells only where there is no secondary mutations, meaning that this retrovirus has integrated into the right place, and then put it back into the cell. So this is the most critical part here. Even taking out the cells from the patient, it is doable. Of course, it is going through this process you know, but this is the most critical stage despite all the investment. Similarly, adenovirus also. Um, Here, the genes which causes disease have been removed. How now? I can question you how the genes are removed. So, of course, by using restriction enzymes. Restriction enzymes is a tool where they can remove the genes which causes viral propagation, and but they need healthy genes that can cure disease. So yeah, retroviruses. And the liposomes, as I mentioned to you, they are lipid bilayer micelles in which the DNA has been encapsulated, and then the bilipid layer could be very much compatible with with the the target cells. The naked DNA where the plasmid can be inserted. The plasmid can be injected into the cells by using microinjection, electroporation, or through chemical means. So, when we are dealing with such experiments, of course, there are risks and benefit both associated. So, of course, a disease can be eradicated, defective cells could be replaced, and this has uh, potential beyond our imagination. Is the you know it can modify human capabilities. When I teach you about of gene editing, then I'll talk about CRISPR. There was a scientist in China who was arrested because he was editing the human baby. I think he was released or his court case was going. So people can edit human baby, and how much that is allowed within the. So again, science also is regulated by government policies, right? That's the chapter we will talk about. And so yes, you develop the technology, but the technology has to be approved by the government, and the government has to take a policy decision to implement that technology, because how much where we are going with this, because this CRISPR technology that I will talk in next class has immense potential to edit human genome. So what are the diseases that can be targeted? Of course, cancer. Cancer, one of the gene is P53, which is a tumor suppressor gene. It is mutated in at least 70 to 80, I think 60 to 70% of the patients. So if we get a healthy P53 gene and it can be inserted into the patient, the tumor cells, the cancer cells, then these normal P53 gene can direct the cells towards apoptosis and the killing of cancer cells. Similarly, cardiovascular disease, in diabetes, the beta cells of the pancreas, they're affected and they don't express insulin. So again, these retroviral vectors can be used to express insulin. Cystic fibrosis, even same for SCID in patients where adenosine deaminase is mutated. Infectious HIV patients also, many other diseases. So particularly, I've been reading that gene therapy is very successful in ocular diseases, in eye diseases. I can't explain why. I have to do more reading, but treatment of chronic eye disease is doable much more than as compared to other soft tissues cancers. Okay, so you have to read these slides, and at this point of time, do you have any question? So if there are no questions, then I leave you early. And I am intentionally not teaching you CRISPR today. I just wanted to bring a boundary because that's more totally different word. And we will learn about CRISPR on next Tuesday, on 11th. On 14th, your midterm is due. Multiple choice questions, cumulative. 1 minute per question, 2 attempt. You are used to it, hopefully. So, any questions?
No question, Professor.
No, Professor.
Okay. If there are no questions, then let me end this class and convert my recording into YouTube and then put it back on the Blackboard. Okay, then bye. Take care.
Same to you.